Thank you. And also, thank you, Sagar and Supratim, for organizing this meeting. It's rare that I get a place to talk about my work because, either it's a biology conference or is a game theory conference. So, yeah. Anyway, so I'm going to talk about. Well, anyway, so I'll talk about the work which I've done in my PhD and also continued parts of it in the last 1 year of my postdoc. Um, it's essentially studying these evolutionary dynamics in spatial structured populations and by using this framework of graphs. And I hope I can convince you probably towards the end of my talk that studying like evolutionary dynamics in spatially structured population can provide some way to steer evolution. So not just look at evolution, how it's happening, but also to control evolution and steer evolutionary trajectory in desired ways. But anyway, so before I talk about structured population, I guess everybody knows, but still. So this is a complete graph, right? It's represents your well-mixed population where there is no structure. So these nodes are asexually reproducing entities, and everybody is connected to everybody else. So everybody can replace everybody else. In the deterministic limit, right, we have seen this replicator equation framework, which if you don't have any frequency dependence, then it's just survival of the fittest. But then you can also incorporate other evolutionary forces on top of this framework. For example, mutation, noise coming from finiteness of the population, right? Genetic drift and some recombination. So if your population is sexually reproducing, and migration and all sort of other forces. But in my talk, I will be focusing on natural selection, mutation, and genetic drift. So the interplay of these three different evolutionary forces and how spatial structure kind of tune the strength of these three different forces in different scenarios. Just to motivate, why like the spatial structured population can generate some counterintuitive results. So this is a model which represents like epithelial skin. So these are the cells which are deep down the skin, and your epithelial is structured in a linear fashion. So this is the outer layer of your skin. So if a cancer cell appears somewhere in between these two extreme cells, it will eventually vanish, like it will die eventually, no matter what's the fitness of this individual. The only way the cancer cell can accumulate in your this epithelial tissue is if it appears in this extreme, like deep down the skin. So making it so in that sense, it's not really about survival of the fittest. So spatial structure kind of nullifies the fitness of these cancer cells. And in that sense, spatial structure kind of it prevents this accumulation of cancer cells. Now, to model spatial structure, as Supratim mentioned, like the name of the framework is called Evolutionary Graph Theory. It was coined in this paper in 2005, and so this is the complete graph, and then we have seen in many other talks in the school. So this lattice, right? It can also be like a lattice, but it can also go a bit more extreme, right? I mean, that there is this one node which is connected to everybody else, the leaf nodes, and everybody else is connected to just this one node. So, and then your links can also be weighted. So the interaction need not be symmetric. So this node can interact like quite a lot with this node, but not the other way. And then you can also have some sort of directedness in the graph. Right. But what do I mean by interactions, right? I mean, so this is about evolution, right? So when we have to talk about some update rule. This also came quite a while in the lectures of Christian, so the modern birth rate update rule. But I want to discuss this in the setting of spatial structured population. What does this update rule looks like? So imagine you have a population at some instance, right? You have these blue individuals and some are yellow individuals. Now, in a given time step, one individual will be selected to reproduce with probability proportional to its fitness. So let's say it turns out to be this yellow individual. And then one of the neighboring individuals, so that's important. That's where spatial structure come into play. So only one of the neighboring individual will be selected for death. So it has 1, 2, 3, 4, 5 neighboring individuals. One of the 5 individuals will be chosen for death at random. And then the offspring replaces the dead individual, and in that way the population updates. You can also have death birth. So in the previous slide, right, it's birth death, but then you can just flip the order of death birth. So what happens in death birth, you take the same population, now you randomly select an individual from the population to die. And then one of the neighboring individual will be selected to reproduce with probability proportional to their fitness. It's this one, and then the population updates. So it turns out this ordering of birth death, it doesn't matter for well-mixed population. But when it comes to these graph structured population, as we'll see, they affect the results a lot. Right, so now I can give you the outline of my talk. I'll present some results on fixation probabilities, right? As you might have guessed already, like there are some absorbing states in the system as the population evolves, but you can also have some mutations, right? Ongoing mutation. So in that sense, then I'll subject these population to some fitness landscape and talk about long-term evolution. But then also your mutation rate can also go beyond this rare mutation regime, as I'll talk about, and then so that will be this mutation selection balance on graphs. And if the time permits, I will just briefly talk about operation on these graphs. Now, just to be on the same page, the fixation probability, so you have a monomorphic population, right? And at some point, the mutation has to appear so that the question of the fixation probability makes sense in the first place. So, but it's not obvious that the mutation will appear on every node with equal probability. So that's what this mutant initialization captures. It tells you like what's the probability that each node is going to be initialized with the mutant. And then the mutation appears, and then there are only 2 fates. So if you run this birth, death, or death, birth, there are only 2 fates in the end. In one case, there is this extinction, and in another case, there is this fixation. So the fixation probability on graph G, so this is the graph, given that the initial mutant appears on node I, and this is the fitness of the mutant, and this is the fitness of the wild type. What do I mean? So the fixation probability in the end, of course, it depends obviously on the graph structure. The initial node on which it appears, the relative fitness of the mutant, population size, right, and of course, the update rule. And this is the average fixation probability on the graph, averaged over all, taking into account all the possible places where the mutant can appear. Yes, please. Oh, yes, by the way, feel free to ask questions during the talk. Oh, you mean some sort of coexistence? Okay, so as long as the population, okay, so first of all, there are no interactions. In the sense, okay, so there are no frequency-dependent interactions that I'll be considering, but even if you consider frequency-dependent interactions, as long as the population size is finite, okay, even infinite, well, yeah, as long as it's finite, there will always be 2 absorbing states under this setup. Right. Now, it turns out these are not 2 mutually exclusive information. And in the next slide, I'll tell you like how update rule governs on which node the mutant will appear. So you start with a monomorphic population, right? At some point, there is a mutation during the birth event. And then the mutant offspring, it moves to the vacant site. And that's how the fixation process can start. So we call this offspring moving, right? Makes sense. But the another possibility is this parent movement. So this offspring, it stays back, but the parent offspring moves, It turns out like there are quite a few studies on this offspring moving rule, right? Like whenever the mutation appears in this graph structured population, those studies always move the offspring, but we just ask, okay, what if the parent moves? And it turns out it can give rise to many other possibilities. So temperature just means that the highly connected node are more likely to receive the initial mutant. And it turns out that with this BDO, like if the offspring move with birth death, then the initial mutant is more likely to appear on the highly connected node. On the other hand, if the parent moves in the birth death setting, then the initial mutant will appear randomly on any of the node with equal probability. So you don't have to remember these things, but I'm just telling you like the idea is like just by the, if this is the update rule, it will automatically govern where the first mutant will appear in the population. Right. So now we can talk about fixation probabilities. So I start with the star graph, and the reason is you can do some maths with it. With other random graphs like it's slightly complicated. But with star graph, you can get exact results for the fixation probabilities. Now the blue one is for the well-mixed population, right? I mean, so this is the mutant's fitness. So these are the advantageous mutant. This is the fitness of the wild type. So again, constant selection, no frequency dependent selection. So it's just a real number value when I say fitness for the individual. And these are the deleterious mutants. Now, right, I guess it makes sense, like as the fitness of the mutant increases, the fixation probability also increases. On the star graph, by the way, this is for birth death, but anyway, doesn't matter. So when the mutant appears on the leaf, or if it's with uniform initialization, then most likely the leaf node will acquire the first mutation because it's uniform mutant placement. In the limit of large n, so n is already large for me, the first mutant will appear on the leaf, and you get this fixation profile. It's called amplifier of selection. Why? Because for advantageous mutant, for the same fitness, if you put the mutant on star graph, it will have higher fixation probability than the well-mixed, and lower for the deleterious sector. So in some sense, this graph amplifies selection. So that's why it's called amplifier of selection. On the other hand, if you do death birth, right? So death birth, the same graph suppresses selection. So that's what I meant by like the update rule affects the fixation probability. And both are uniform initialization. So we just change the update rule. And for temperature initialization, meaning if this node, if the initial mutant starts from the highly connected node, then for the same update rule, you see this highly, like the fixation probability is quite low, regardless of the mutant's fitness. And for this one, you get something like this profile in the death birth sector. Now, you can forget about all this, but the only thing I want to take away from, take away from this slide is this profile. So if you notice something here. So I was new to evolutionary biology, and whenever I would talk to population geneticists, they would always tell me that, okay, it's always the beneficial mutations, which kind of is relevant for evolution, at least in simpler scenario. But this is but here we see something interesting that you get, even in the limit of large population size, there is an example where you have non-zero probability of fixation for deleterious mutations. So near neutrality, it's almost half. And for all other settings, you see that the probability of fixation in the deleterious profile, it goes to 0 as the population size increases. And yeah, I mean, we have some results for in the limit of large and what this fixation probability is for this orange curve. Now, this is just for star graph, right? You may ask, okay, what about other graph topologies? And it turns out we recover the same results. So how to read these plots? So you can focus on, let's say, this one. So we generate random graphs of size 8, small, but we generate, and with different probability of link connection. So we generate the Edo-Shreni graphs. And most of the graphs are these amplifier of selection under this updating, which was the case for star graph also. So whatever we saw for the star graphs, it turns out most of the random graphs have the same fixation profiles. And so the most of the graph have this amplifier of fixation where you have higher probability of fixation for deleterious mutations. Now, although we couldn't derive any general result for the selection, but under neutral dynamics, we have some results. So this is the probability of fixation for neutral. If your mutant is neutral, right, the fixation probability is 1 over n. We know it's the case for well-mixed graph, but this is true for any graph. It's 1 over n. But for parent movement, that's not true. It's not equals to 1 over n. And in fact, we have shown that under parent movement, for any graph, the fixation probability will be greater than the fixation probability of 1 over n. So if you take a neutral mutation, if you have this sort of updating, the fixation probability will always be greater than 1 over n. And in some cases, it can be independent of n. Right. And the reason it happens is because under parent movement, you tend to put mutants in more of highly connected nodes, as you saw for star graph. And this we saw to be consistent with other graph topologies also. And in this plot, you can see what's the increase in fixation probability when you go from this offspring movement kind of update rules to parent movement kind of update rule. So the increase can be as much as 10 to the power 4 in terms of fixation probability. Right, so the summary so far, like for this section is like, okay, the parent movement, we introduced this parent movement scheme and which gave rise to this new profile where you have non-zero probability for deleterious mutation in the limit of large n. Right, so I think the way you can think is, so let's say the mutation appeared here and it's death birth, right? So in the next step, time step, one of the individual will be selected for death and it's most likely going to be a leaf node. But the leaves are only connected to center. So the center will send their offspring. So as long as this mutant stays on the center, it will kind of take over the population. But it's not strictly one because at some point the center also changes. So, but you can probably convince that yourself that, yeah, it works out that it will have higher fixation probability than the other two cases. Exactly. Right, but overall what we saw, so in the previous slide, right, I mean, the difference is low for this Erdo-Shreni graph, which is, like there is not much difference between offspring because it's almost close to well-mixed, but you need to have some sort of heterogeneity in the degree distribution. So for example, for Baravasi, Albert graph, like you see a significantly higher difference because you have this hub-like of structure, right? I mean, so yeah. Right. Exactly. Right. 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 So, right. So yeah. So this is the fixation probability for parent movement, right? So this is the weight matrix that enters the relation, and right. So there are some results in the literature and using some arithmetic mean. Sorry. Yes. This is for any graph, any weighted graph. Right. So now let's just switch gear, and now the question is, okay, what happens if like you have subsequent mutations? So then the idea is, okay, again you have population, right? Oh, sorry. You have a population, and then at some point during something like with probability mu, there will be a mutation, and with 1 minus mu, there won't be any mutation. But if there is a mutation, the fitness of the mutant will be selected from a line with some distribution, which is missing here, but anyway, not important again. Yeah, it comes back. So in the limit of weak mutation, the dynamics is essentially a random walk on this very simple fitness landscape. So for example, the population is monomorphic, then there comes a mutation, And the fitness of the mutant is sampled from this distribution, which is centered around parent's fitness. So this is the fitness of the mutant. And then it also has to fix the mutation. That's only the population can move to another point, right? And then, as you can imagine, this will be a Markov chain. It's a Markov process. And you can write master equation, and assuming some reversibility in the process, you can solve for the detailed balance. And the thing to notice is that the steady state solution, so what does this mean? This is the probability that your graph G in the long term at fitness value F. So this depends only in the ratios of fixation probability, which makes sense. Right, so this is the result which was for fixation probability. Now what happens in the long term? So on the x-axis is the population size. On the y-axis is the average fitness attained by this population in the long term. So if you just look at the blue one, so it expected that as the population size increases, selection becomes stronger, you tend to accept beneficial mutations more easily, hence the fitness increases for the blue one. For the green one also makes sense because the fixation probability to increase beneficial mutation is higher than the blue one, right? So it has higher fitness in the long term than the blue one, which is a well-mixed population. But what was surprising for us was this one, that this pinkish curve, which has almost zero probability to accept beneficial mutation, yet it attains not only higher fitness than the well-mixed population, but almost the same as the green one. And the reason for that is that it's good at rejecting deleterious mutations than other graphs. So it's not only important how good you are in accepting beneficial mutations, right, which is the usual consensus, but it's also important how good you are in rejecting deleterious mutations. So in PopGen literature, I mean, if you're just focusing on well-mixed, you'll never see something like this playing into the long-term fitness. But here it turns out, even these are very small probabilities, they tend to, in the long run, affect the long-term consequences. Right, and this we found to be true for other random graphs also. So the result is not just true for star graph, but for other random Evolutionary graphs. Same thing with the death birth dynamics, right? So it makes sense, like the blue one, the well-mixed has higher fitness than the red one because it's not good at accepting beneficial mutations. But the orange one has the lowest fitness because, fine, it has higher probability to accept beneficial mutations, but it's also accepting deleterious mutation with very high probabilities. So yeah, so that's the summary that, okay, rejecting deleterious mutation is also important as fixing beneficial mutations for adaptive evolution. Now we were focusing in the weak mutation sector. So this point corresponds to just fixation probability, right, when mu is 0. But if mu is finite and yet very small, like mu smaller than n square inverse, then you are in the weak mutation regime. But what if you are somewhere here? And then just move to this mutation selection balance on graphs. So just to set the stage, so it's very simple system, right? You just have two types, wild type and mutant. Wild type can mutate to mutant with probability mu, mutant can mutate to wild type with probability mu. In the steady state, so the frequency, if your mutant is fitter than the wild type, whose fitness is 1, the frequency of the mutant is not 1, right, because of finite mutation probability. So that's the mutation load, which mutation puts on the population. And you can see it from this figure. So if your mutant is beneficial, there is this, it's not suddenly jumps to 1 because of finite mutation rate. And for high mutation rate, there is this reversal in this fixation profile. So the deleterious mutation, there will be found in abundance as opposed to beneficial mutation. But that's only for high mu, very high mu. But this is for well mixed. Now we asked, like, okay, what happens for these degree heterogeneous graphs? So we ran some simulations. And the first observation is, no matter whether it's parent movement or offspring movement dynamics, you'll always have the effect of mutation will be higher. So the mutation will be stronger as compared to well-mixed population in these degree heterogeneous graph, and the effect is even higher for parent movement. And that effect increases as you go to more and more degree heterogeneous graphs. More specifically, if you look at this Barabasi-Albert graph, the dynamics is almost neutral. The result is almost 0.5. So no matter what's the fitness of the mutant, the long-term frequency of the mutant will be close to 0.5. And which can mean that these kind of population with this sort of updating can maintain high diversity regardless of individual's fitness. So even if due to my genes or whatever I have very high fitness, just because of the population structure, I will behave neutrally. So to see these things in detail, we move to star graph with this parent movement dynamics. And turns out that the steady state frequency of mutant on this star graph, it depends on the fitness of the mutant, no surprise, but it depends on the product of mu and n. So in well-mixed population, it will be a function of mu and f. N will disappear. But here, it's the product of mu n. So there is this scaling of mu to mu n on these graphs. And that's what we call amplification of mutation. So the natural selection is kind of crushed. I mean, the mutation is so high in these graphs. So even if mu is small, like mu itself is small, but just when it's coupled with n, it's very large in these populations. And we derived this result that in the steady state, the frequency of the mutant will be 1 over 1 plus f, where f is mutant's fitness relative to wild type. What this says, if the fitness of the mutant is infinite, its frequency in the steady state will be 0. And if its fitness is 0, its frequency will be 1. To see it in pictures, it's a complete reversal of selection. Higher the fitness of your mutant, lower will be its proportion in the steady state. And lower its fitness, higher will be its abundance. And we just call it survival of the weakest. Right. So the summary is, okay, overall, like you can forget about death, birth, offspring, parent. The summary which is coming out is, okay, that you cannot ignore really deleterious mutations. It seems to be playing role in the fixation regime when mu was small, right? You have this non-zero fixation probability, a very large population. And it also playing role in this when the mutation rate is outside weak mutation regime. So, and I guess this is just the last slide. Okay. So this is a known result. So if you have this double star graph, if you look at the fixation probabilities of cooperators and defectors, so playing simple, like playing donation game, the cooperation will be favored over defection on this double star graph under this offspring movement rule. But under parent movement rule, true cooperators are again favored over defectors. But if you look at the scale of y-axis, it's almost 10 times higher, the probability of fixation, than in this standard case. Right, so I like to thank my co-authors and the generous funding and from MPI and the Department of Theoretical Biology at Bloom. So thank you.
So regarding this mutant initialization,
right?
So you are saying that sir, every node has a probability with which. So why are you taking into account that only one node is getting initialized?
Right. So okay, this is not a good way. Right. I mean, the short answer is like, we are interested in fixation process, and generally, the most conservative case is, okay, the population is monomorphic and it's just one mutant, right? That has to appear somewhere. So you start with just one mutation. But you can also ask question, okay, what's the probability if you start with 2 mutants or 3 mutants?
No, because the rest of the nodes have non-zero probability, right?
Yes. Every node has a non-zero probability.
Yeah. Then it's more
But-
practical.
Right. So, sorry. Yeah.
It's more practical to take more than one.
Well, so the probability, what I mean by probability, it's like, okay, what's the probability that if you have to put one mutant in this population, what's the probability of this node, this particular node getting that mutation? Yeah.
One more question is, you are taking, at one point of time, I saw the fitness to be a realization from some distribution.
Right.
So why is it so? Like,
Yes. So the short answer is again, like if you are somewhere here as a parent and there is a mutation, so this is fitness, it makes sense that the fitness is sampled close to you, right? I mean, the offspring, there is some correlation with parents' fitness. So you sample it with some distribution. But
it happens like that?
yes, so good point. So we considered many distributions which are mentioned in the literature. So we from uniform to Gaussian, and the results are robust to those distribution, the choice of distributions.
And one more thing is, when you brought into account a mu,
Right.
so I want to know, is there no, so mu is the probability or is the rate from wild type to mutant type?
Right.
So is there no relation between mu and the fitness? So higher the fitness, more will be mu. Is there no such relation?
No, in the model we didn't consider like fitness. We kept it independent. Mutation is like we kept them as like independent forces, and then we just asked, okay, they are independent. How will they manifest like in the long term when they interact with each other?
Can exist?
I'm sure, like, I mean, yes, like there are always, like these forces, mutation rate is also governed by natural selection, and then there's always some feedback. So yeah, but we're just like looking at some small model. So we make some assumptions. So, yeah. And these are standard assumptions. So